So the map originally contained, the map that Brodmann came up with here, it originally contained 52 areas. Now we are up to 200 and counting. So with so this is kind of just a clearer view of the Brodmann's areas, and of course, you know, we've got them on both sides, right? So so yeah, there's a lot, you know, but you guys don't have to know all these areas, certainly not. So what can we say about Brodmann's classification? Just 1 second. Well, just 1 second. So with Brodmann's classification system, it may seem unsystematic. You know, like there's just numbers everywhere, right? So it's true that Brodmann's numbering system had more to do with the order in which he sampled the region than any meaningful relation between areas. So nevertheless, in some regions, the numbering system does roughly correspond with relations between areas that carry out similar functions, such as vision. You know, so vision, we've got, let me just flip back. So with vision, we've got 17, 18, 19, right? So these are all visual areas. You know, you've got, you know, 1, 2, 3 up here, 4, 5, you know, being, you know, kind of the motor cortex, somatosensory cortex, all being kind of grouped together. So in some cases, the numbering does correspond with similar functions, but most of the case, it's not related to similar functions. So really, the labeling of the nervous system is not fully standardized. So you can have, you know, different names for the same region. So a brain region may have a Brodmann label. It may have a psychoarchitectonic label. It may have an anatomical label. It may have a functional area label. So, you know, and a really good example of this is the visual cortex. You know, so it's got a bunch of different names, right? So it's Brodmann's area 17. It's also called the striate cortex because of the way that it stains. It stains in a striped or striated pattern. So, you know, kind of from a cytoarchitectonic perspective, it's striped. It stripes in a striped pattern, so it's called the striate cortex. It's also called, you know, the anatomical label is the calcarine sulcus, or sorry, the calcarine fissure. And it's also called V1, primary visual cortex. So a lot of regions can have different names depending on how you're looking at it, whether you're looking at it anatomically, functionally, cytoarchitectonically, from Brodmann's area. So it can be complicated when you're trying to label something. But we're not going to get too hung up on these different labels here. I'm more a fan of just calling things, you know, V1, visual cortex, M1, motor cortex. So couple of other divisions of the cerebral cortex that are much easier to follow than any of those ones. Neocortex, allocortex. So, so with neocortex versus allocortex, so this is considered, all of this white region here is considered neocortex, and this blue region is considered allocortex. And the distinction really between these two types of cortex is their evolutionary history. So basically, neocortex means new cortex. So it's named as such because it is evolutionarily newer, whereas the allocortex is evolutionarily older. And allo is, I believe it is Latin for other. Neo is Latin for new. So we've got new cortex, we've got other cortex. So really just the distinction here is, when they developed evolutionarily. So the allocortex, that contains like regions like the limbic system and the basal ganglia, which we looked a lot at last class. And neocortex, evolutionarily newer. So let's look at the neocortex. So cells in the cortex are divided in a variety of different ways, different labels, different names, different structures, but basically the kind of two broad divisions of cortical cells is the presence or absence of dendritic spines. So the dendritic spines, I just flip ahead and I'll show you. So this right here, if you look at this right here, these are spines on a dendrite. So before we move to that, let's just flip back. So divided based on the presence or absence of dendritic spines. And dendritic spines are important for increasing the surface area of the cell. And dendritic spines tend to be excitatory. So using neurotransmitter systems that are excitatory, like glutamate, for example. So whether or not a cell has dendritic spines or not has a certain name. So if a cell has dendritic spines, it's called a spiny neuron. So we look at this picture out of your textbook. So it's got all these color-coded here down in the legend. So spiny, let's look for some spiny stellate cells. So up here. So spiny cells, spiny neurons, they have dendritic spines. So a couple of examples, pyramidal cells and stellate cells. So these are both classified as spiny neurons because they have dendritic spines. So pyramidal, stellate, what does that mean? It just refers to the shape of the cell body. So pyramidal cells, they have long pyramid-shaped cell bodies, and they also have long axons, right? So this is a pyramidal cell, this is a pyramidal cell, so it's got a triangular shaped cell body, and they've got long axons. Stellate cells, they're also classified as a spiny neuron because they have dendritic spines, but they are star-shaped. So if we look up here, for our spiny stellate cells, you can see they're kind of star-shaped. So that's what they get their name from, basically getting their name from their shape. Pyramidal cells, shaped like pyramids. Stellate cells, shaped like stars. So stellate cells, unlike pyramidal cells, they have short axons. So stellate cells are also classified as an interneuron, meaning that basically you can think of interneurons as communicating within a tiny region, kind of in a tiny bubble, because they have shorter axons, right? So if you look up here with these stellate cells, you can see that they have very short axons compared to this pyramidal cell. So stellate cells, they're small interneurons, and they have short axons, which means that they communicate in their tiny little region of the cortex. So that's basically what an interneuron is. It's a type of cell that communicates within its tiny little space and has either short axons or no axons. So in the cortex, we've got stellate cells, which are a type of interneuron. Okay, so what about these other ones? A-spiny neurons. So A-spiny neurons, they are neurons that don't have those dendritic spines. So they can also be interneurons, just like the stellate cells are a type of interneuron, but stellate cells are interneurons that have dendritic spines. A-spiny neurons, they are also interneurons, but they have no dendritic spines. So let's find some of these A-spiny cells in our legend. So peach ones. So here. And these ones also have, they've got a lot of different, there's a lot of different types of A-spiny neurons, and they are usually labeled for their shape. So we've got a basket interneuron here. So it's a type of A-spiny neuron, and they're basket shaped. So we've also got other types here, double bouquet A-spiny neurons. I guess they kind of look like two bouquets of flowers pointing the opposite way. These things, they've got these neurons, they've got a lot of different names, mostly based on what, anatomists think that they look like. So anyway, many different types, commonly named for their appearance, but the key thing is that they don't have these dendritic spines. So if you look up here, this pyramidal cell, it's got the dendritic spines. If we look at our spiny stellate cells, they've got their dendritic spines. But if we look down here at these ones and these ones and these ones, they don't have those dendritic spines. So that's one difference between spiny and A-spiny neurons, is that they don't have those dendritic spines. But the other difference is where spiny neurons tend to be excitatory, A-spiny neurons are inhibitory and typically use GABA as their primary neurotransmitter. So there can also be other neurotransmitter systems occurring with them, primarily using GABA as their neurotransmitter. Okay, so I'm just going to flip back because we've got a couple of other terms on this figure. It's a busy figure. So let's look at what this means. So we've got afferents and efferents. So let's look at what that means. So afferents and efferents, really just terms that tell you where the information is flowing from or to. So afferents, this refers to axons that are bringing information into a structure. So if we look at this figure here, afferents, and we've got our flow of information. So you can think of this, we've got these numbers here that we haven't gotten to yet, so just ignore those for now. But let's think about this as being the inner part of the cortex that's laying up against those subcortical structures, and this being the outside. So with the afferents, these are projections that are bringing information up through the cortex into the cortex, and then we've got efferents. So efferents, information flowing the other way. So with afferents, we've got axons that are bringing information into a structure. With efferents, we've got axons that are bringing information away from the structure. So afferents tend to be sensory neurons, and efferents tend to be motor neurons. So if you look at the motor cortex versus the somatosensory cortex, a lot more, you're going to have a lot more afferents in the somatosensory cortex, a lot more efferents in the motor cortex, which we just spent a lot of time talking about all those descending motor pathways coming from motor cortex and other regions. So that would be these efferents. Okay, so let's look at these numbers, what they mean. So the cortical layers have, we've got the neocortex has between 4 and 6. Your textbook says 4 and 6 layers, tends to be more like 6. So we've got 6 cortical layers, labeled with Roman numerals just to make it extra confusing. But this is 1, 2, 3, 4, 5, and 6. So 6 layers. So like I said, you can think of this being the outside of the cortex and this being the innermost of, here, let me just flip back to what I think will be more useful to look at. Okay, so you can think about layer 1 being here, layer 2 being here, 3 and 4 and 5 and 6. Okay, so 6 is going to be the layer pushing up against this white matter, and layer 1 is going to be the layer on the outermost surface layer. So layer 1 out here, all the way into layer 6, which is going to be the layer pushing up against this white matter. Okay, so let's flip back to our layers. Okay. So each of these layers have different functions and different inputs and different outputs. So we've got specific and non-specific afferents. So with the specific afferents, these are afferents that carry information to the cortex and terminate on a small number of cortical regions, versus non-specific afferents, which also carry information to the cortex, but they carry it to a large number of cortical regions. So you can think specific afferents having kind of a narrow area that they're terminating on, non-specific afferents having a large area that they could terminate on. So, so this figure here, we can come back to this. I've got this legend on here now with S and NS indicating specific and non-specific afferents. So you can see with the non-specific afferents projecting to a larger region, larger area, specific ones projecting to just one area. Again, in this case, projecting only to levels 6, 5, 4. And then we've also got some other labels on here, the cortico to cortical, afferents, cortical to subcortical, cortical to spinal, cortical to thalamic, cortical to thalamic. So I'm going to discuss these different, you know, these connections to each of the layers with words instead of using this figure. So we'll get to what the projections for each of these layers look like here in a minute. Okay, but first, okay, we've got our neocortex, we've got our 6 layers, and I've got them numbered here with regular numbers for those of you who don't like Roman numerals. So how are these 6 layers determined? How do we know there's 6 layers? So the neurons of each layer are typically similar in appearance within that layer, and different between the layers. So you can clearly see this with a Nissl stain or a Golgi stain. You know, so even if you were to kind of take out these numbers, right, even if you didn't even look at them, you can still kind of tell just from these stains that these look like they belong together. These look like they belong together. These look like they belong together. These do, and these do, although it gets a little less clear the more superficial layers. But basically, the neurons in each layer, they are grouped into a layer because they are similar in appearance. They appear similar when they're stained. So with these 2 different types of stains, this is actually showing the same thing, right? So this is just a Nissl stain, a Golgi stain, with the Nissl stain showing the cell bodies in this one, and the Golgi stain showing axons and dendrites, more so than cell bodies. So within each of these layers, so we've got similar cells. So in, for example, layer 5 and 6, we've got a lot of pyramidal cells in layer 5 and 6. Layer 4, we've got a lot of stellate cells. So these are going to end up being those stellate cells that are star-shaped. Layer 3, we're back to pyramidal cells. Layer 2, we've got a mixture of both, probably more stellate cells. So there's different types of cells in each layer. So also with these layers, they develop and mature over the lifespan at different rates. So the deeper layers, layer 5 and 6, these layers mature earlier during gestation, so in utero, and 5 and 6 project, primarily to targets outside of the cortex. So these are your motor regions. Layer 4 is typically a sensory information layer, an input layer. So let me just flip ahead to some better slides that explain this. Okay, so neurons within a layer, they are similar. Neurons between are different between the layers. Okay, so let's look at this with a little bit of a simplified view of the layers with regular numbers. So layers 5 and 6, these layers have more pyramidal cells. So they have more pyramidal cells, and these are deep output layers. So layers 5 and 6 send information to subcortical regions. And like I said, layers 5 and 6 mature earlier on in the developing nervous system compared to layers 4, 3, 2, and 1. So layer 5 Layer 5, considered an output layer, and layer 5 sends um, a lot of information from the cortex back to the thalamus. So if I flip back again to our figure here, so we've got our CT label, cortical thalamic efferents rising from layer 6. Okay, let's skip ahead. That's confusing figure. So layer 5 and 6 tend to be output layers and mature earlier and have more pyramidal cells. Okay, what about layer 4? Layer 4, quite different from layer 5 and 6. So layer 4 has many stellate cells, and it is an input layer. So it's an input layer which is going to be where the axons from the thalamus and other subcortical regions are sending their information to. So layer 4 has a lot of stellate cells and receives its information primarily from the thalamus and other subcortical and cortical areas. Okay, so layers 1, 2, and 3, they are considered the superficial layers, and they do mature last developmentally. And layers 1, 2, and 3, a little bit less specific in their input-output pathways. So they don't directly receive sensory or motor, they don't directly receive sensory input or direct motor output. And they are suspected to participate in higher cognitive functions. And less is known about these layers at this point compared to the other ones. So the thickness of each one of these layers, so we've got just a section here showing a certain level of thickness, but this thickness doesn't, it's not uniform across the whole cortex. And the thickness of each layer depends on its function. So the thickness of each layer varies across the cortex depending on the function of the particular region. So let's look at this. Okay, so we take, so this is our section of motor cortex and our section of somatosensory cortex that we've been looking at. So let's look at the thickness of each one. So as you can see, if you look at the column that's here for the motor cortex versus the column for the somatosensory cortex, you'll notice that the thickness of these layers is not the same. So layer 1 and 2, the same. But if you look especially at layers 4 for the somatosensory system, it is a lot thicker over here for the somatosensory system than it is over here when we look at layer 4 for the motor cortex. So layer 4 has mostly stellate cells, which are those cells that receive sensory input. And like I said, layer 4 is primarily a sensory input region. So, regions like the parietal lobe and the occipital lobe, they are predominantly sensory areas as well. So they have a thicker layer 4. So somatosensory cortex, parietal cortex, visual cortex, these regions are going to have a thicker layer 4 compared to motor cortex. So motor cortex, on the other hand, it has a lot of pyramidal cells, because remember, the pyramidal cells are those ones that have those really long axons. So we know, based on what we've already talked about in the class, that the motor system sends these really long projections, down from the cortex and, down through the brain stem, and onto the spinal cord. So pyramidal cells send signals, from the cortex, the brainstem, the spinal cord, for the purpose of directing voluntary movement and involuntary movement. So layer 5 in 5 and 6 in the motor cortex is a lot thicker. Layer 5 and layer 6 in the motor cortex is a lot thicker compared to what you see in layer 5 and 6 in a sensory region. So these layer 5 and layer 6 tend to be made up of those pyramidal cells that have those long axons. So considering the functions of the motor cortex, it's not surprising that layer 5 and 6 is going to be really thick in the motor cortex. So somatosensory cortex, going to have thicker layer 4. Sensory region is going to have a thicker layer 4. Motor region is going to have a thicker layer 5 and 6. So thickness varies across the cortex, and the thickness of each layer depends on the function of the particular area that you're looking at. So in addition to layers, we also have another type of organization we see in the neocortex, and that is a column arrangement. So neurons in the neocortex are arranged vertically in columns. So these columns, they run perpendicular to the surface. So you can think the surface of the cortex, and these are the columns running perpendicular to the cortex, to the surface. So these columns are sometimes referred to as mini columns or microcolumns, and they are arranged. They are arranged in such a way that the neurons contained in one column respond to similar properties. So if we look at this column, let's say this column, let's say we're looking at the visual cortex, which is what we're going to look at next. But let's say, this column here, we're looking at the visual cortex. All of these cells would respond to one particular type of orientation. So one particular kind of visual input. So say all of the cells in this column, they respond to oriented lines that have a vertical, completely vertical 0-degree orientation. So that would mean that the cells here in this layer would respond to a completely straight vertical line, and so would these ones, and so would these ones, and so would these ones, and so would these ones. So, the columns, neurons within these columns, they synapse with those from the layers above or below them, forming this kind of little circuit that functions as a unit. So these layers will be getting their input from down here, and then from up here, and so on. So the neurons within a column, they receive input from the layers above or below them. And this column arrangement is a kind of a fundamental type of organization in the cortex, this columnar arrangement. So within these columns, how big are they? Well, these columns, they can contain anywhere from 150 to 300 neurons and are about between 0.5 millimeter to 2 millimeters wide. So again, depending on the area of the cortex, but pretty tiny, pretty tiny little columns containing anywhere from 150 to 300 neurons. So also, this is an estimate, that could change with, advancements in microscopy. So that's the current estimates, but again, we are at the mercy of technology when it comes to understanding these things. So this kind of gives you a little bit more of a realistic, perhaps, perspective on this column arrangement. So each layer does have horizontal, vertical, as well as horizontal connections. So you can really see that a little bit better in this figure. So we've got the vertical flow of information within a column, and then we've got some horizontal communication between columns. So the vertical information sends vertical connections send information within the column, horizontal connections send it between the columns. And this column organization, it's certainly more prominent, more apparent in primary sensory areas, and perhaps less clear in other regions of the brain. So let's look at this column arrangement in one region where the column arrangement is very prominent, and that is V1, the primary visual cortex. So columns are an important type of arrangement that's seen in the primary visual cortex. And these columns are organized into what are called orientation columns. They're called orientation columns because the cells within a column respond to a similar orientation. So cells within a column in the primary visual cortex will have similar orientation preferences. So remember when we talked about in chapter 8, we talked a little bit about receptive fields in the visual cortex and things like that. So we already know that cells in the visual cortex respond to orientation, and the receptive fields in the visual cortex, cells in the visual cortex have a receptive field that corresponds to orientation of lines, orientation of stimuli in the environment. So in these columns, cells that have a similar orientation preference, so for example, cells that respond to a 0-degree orientation or a completely vertical line, they will be arranged in a column. So that's one type of column arrangement in the visual cortex, but there's also another. So columns in the visual cortex are organized according to orientation preference and also according to the eye that they have, the eye that they respond to. So these are called ocular dominance columns. So cells in these columns respond to input from the left eye or the right eye, and they're also arranged into a column based on the eye that they prefer to respond to. So we're going to talk a little bit more about this just to kind of clear up this. That was just kind of an overview of the two different kind of column arrangements. So let's look at what orientation and ocular dominance columns look like in the primary visual cortex. So cells in V1, primary visual cortex, are organized into columns with cells that have the same orientation preference being grouped together into these orientation columns. And this is organized in a it's very orderly, this organization. And it shifts basically these columns and the columns and their orientation preference shifts just slightly as you move around the visual cortex. So for example, if we can imagine that this is the surface of the cortex and these are our layers, and we were to take an electrode and insert it and penetrate all of the layers of the primary visual cortex, this is what you would get. You would get cells here that have a preferred orientation that they respond to of a completely horizontal line. So if you extended the electrode to here, these cells would respond to a completely horizontal line. And then if you extended it further down, these ones down here would also respond to the same horizontal orientation. And then down further, same orientation preference, same orientation preference. If you shift over a bit and then you start inserting your electrode this way, they're going to have a slightly different but orderly progression of orientation preference. So now these ones are going to respond to this orientation. And then if you insert the electrode in a little further, this orientation, and still all the same orientation. So neurons within these columns, they fire maximally to the same orientation. And then adjacent columns change their preference in an orderly fashion. So if you inserted your electrode here, it would be slightly different than this one, and then slightly different over here, and slightly different. Okay, so orientation, columns in the visual cortex organized based on orientation preference. Like I said, we've got two types of columns. We've also got ocular dominance columns. So ocular dominance columns refer to columns of neurons that tend to have a preferred eye. So by the time we reach V1, we've got inputs from both eyes being combined. So again, if we think back to our visual pathways that we covered in chapter 8, at this point, the input from both eyes has been combined. And neurons in the primary visual cortex, they can respond to input from both eyes, but most neurons in the primary visual cortex have a preferred eye. So yes, they can respond to input from both eyes, but they respond more vigorously to one eye over the other. So this is referred to as ocular dominance. So ocular dominance is a neuron's tendency to respond more strongly to stimulation in one eye compared to the other. So this actually, you can see this pretty clearly in different types of staining. So this is the visual cortex of a person who lost an eye. So this is, of course, post-mortem. So this is an autopsy situation. So this person, they lost their eye. So this means that the person had lost their eye, one eye before they died, so that the neurons were only responding to only the neurons from the other eye, the only neurons that respond to the other eye that was intact would be active at the time that the person passed away. So, so their brain was stained after they passed away with something type of material, type of enzyme, stained for a type of enzyme that is used in cellular metabolism. So this means that the stain would show up and show you the cells that had been recently active at the time the person died. So only neurons responding to the intact eye would be active because they didn't have another eye. So these neurons that were active, showing metabolism, would show up dark on the stain. So that's what we're seeing here. So these have been stained. This visual cortex has been stained with a material that indicates cells that were recently active before the person died. So you can see these ocular dominance columns being shown with these light and dark stripes. So only the neurons that were active are going to be showing up dark, and the light spaces are going to be, the columns corresponding to the other eye. So the dark and light stripes of this patient show the ocular dominance columns of this person. Nicely, because they were only receiving input from one eye. So we have kind of another level of organization as well in the visual cortex that combines orientation, preference, and ocular dominance. And this is called hypercolumn organization. So hypercolumns incorporate the column organization and ocular dominance the column organization for orientation preference and the ocular dominance organization. So let's just look at this. So what we have here is 3 ocular dominance columns. So you can see, so we've got an orientation column in purple. We've got an orientation preference. So this column's preferred orientation, and then slightly different here, slightly different, in this very organized fashion representing all orientations for the left eye. And then beside it, we're going to have another column that also responds to this orientation and this orientation and this orientation and so on and so forth, but for the right eye. And then we're going to have another column. So that's this is one hypercolumn. So we've got an eye preference, we've got an orientation preference. So we've got this orientation for the left eye and the same orientation for the right eye. So we've got a hypercolumn, which is basically combining an orientation column and an ocular dominance column. So this is one hypercolumn. This is another hypercolumn. This is another hypercolumn. So they all contain preferred orientation and with it being incorporated with a preferred eye. So we've got a section that corresponds to all orientations in the left eye and all those orientations in the right eye. And we've got hypercolumns that represent all locations on the retina. So we can think of this as being about a 1 millimeter section of the visual cortex that represents, in these hypercolumns, all possible orientations in one location of the eye. One set has a preferred one set prefers the left eye, the other prefers the right eye. Okay, so we've got about a 1 millimeter section of visual V1 that represents all possible orientations in one location in the retina. And then we've got one set preferring the left eye, the other preferring the right eye. And the transition between this orientation preference and eye preference, it's gradual. So it's gradual. So it's not a completely black and white distinction. It's a gradual change between, so between this column and this column, you know, this column here might respond to a slightly different orientation, but just better to this orientation. So it's a gradual transition between orientation and eye preference.